Well, thanks, Corey, for the introduction, and thanks all for coming. So I was asked to give a brief talk on some of the recent advances in CRISPR technologies. So as you all know, this is really an immensely exploding field. There are a lot of publications. The field really has been advancing on multiple fronts across basic biology, biotechnology, and also medicine. But today, I really wanted to highlight 4 different aspects of Cas9 applications. And so at the outset, the first thing I'd really like to emphasize is that the CRISPR field really came out of a beautiful story of basic biology. So just as recombinant DNA technologies really benefited from basic research into the restriction enzymes that govern warfare between phage and bacteria, similarly, the latest generation of Cas9-based genome engineering tools came from similar research into this anti-phage microbial defense systems. CRISPR, of course, is originally an adaptive immune system found throughout bacteria and archaea, and it allows a host cell to defend itself against invading bacteriophages or other mobile genetic elements. Over the past couple of years, there has been a tremendous amount of work through reverse engineering of the CRISPR-Cas9 system and the heterologous expression into various cell types, allowing us to really generate a simple and universal programmable nuclease that allows us to generate targeted mutations in virtually any organism of interest. So if we take any short stretch of genomic DNA, as shown here, in whatever cell type you like, you'll find that there's this PAM motif, this short stretch of DNA for the S pyogenase Cas9 that's typically used by genome engineers. That's an NHEJ motif that is recognized by the Cas9 protein. So Cas9 recognizes this PAM, docks down onto the DNA, and then interrogates for sequence complementarity between the target DNA immediately upstream of this PAM motif and a 20 nucleotide guide sequence found on a guide RNA that's required for targeting. So this really requires a minimal 2-component system, the Cas9 nuclease as well as the guide RNA that will bring the Cas9 complex and assemble it at the genomic target of interest. Once Cas9 recognizes the PAM and interrogates for sequence complementarity, it basically makes these 2 checks and then activates its 2 nuclease domains, known as RubC as well as H and H, which in a concerted fashion nick the top and bottom strands of the DNA to generate a blunt-ended double-strand break in the genome that can then be repaired by endogenous repair pathways in the cell, typically non-homologous end joining or homologous recombination. This allows us to introduce, of course, targeted mutations in the cell type and the genome of interest. So this is all under the general umbrella of targeted DNA breaks facilitating alteration of the genome, where generating a DNA double-strand break, this could be via EMS mutagenesis, UV irradiation, tissue culture, or programmable nucleases like Cas9 or previous talons and zinc fingers, you essentially activate an SOS response in the cell that will then typically go through 2 major repair pathways, either the non-homologous end joining pathway, which leads to the introduction of short indel insertions or deletions at the break site that eventually leads to gene loss of function, or the more precise but much lower frequency homology-directed repair or homologous recombination process, which is a much more precise repair pathway that can lead to the introduction of point mutations, fluorescent reporters, and so forth. And we'll come back to this further. So the Cas9 system has really come to be an astonishingly versatile tool that can really engineer biology in various creative ways. This, of course, is the classical nuclease, where you generate a blunt-end double-strand break to activate the NHEJ or HR in cells. But if you catalytically inactivate one of the 2 nuclease domains within Cas9, you can generate 2 different nickase mutants. So this is interesting because this allows us to generate more precise mutations when the nuclease Cas9 may have certain kinds of off-target effects. Because single-stranded breaks in the genome are typically repaired with much higher fidelity, yet still allows strand invasion and initiation of recombination or various linear donors, this allows us to do more precise HR. Similarly, you can engineer Cas9 to become a double nickase. So by having 2 Cas9 complexes assembling at the same DNA locus within close proximity, by nicking the top and bottom strands at the same time, you can mimic this overhanged double-stranded break in DNA to introduce more precise DNA double-strand breaks on target. Again, Cas9 can be applied in a variety of ways across cell types in animals. It can be directly transfected or nucleofected or electroporated into cell lines. It can be directly injected into zygotes for generating animal models for disease and various biological processes. And finally, it can be packaged into viral vectors such as adeno-associated virus, pictured here, lentivirus, adenovirus, and so forth, for the direct modeling of your biological question directly in somatic tissues. We can also adapt Cas9 for high-throughput functional screens. So by generating an oligo chip that contains tens to hundreds of thousands of individual guide RNAs, you can clone them and pool into a lentiviral library. If done properly, this lentivirus library containing tens to hundreds of thousands of unique guides can then infect a population of cells. If you do this in the right way such that you get an individual infection event per cell, in one dish you can generate a kaleidoscope of genomes that can then be screened for your phenotype of interest. Finally, Cas9 can also be catalytically inactivated. When you generate the nuclease mutant, you inactivate one of the two nuclease domains. If both of those are mutated, you can convert Cas9 into a generalized DNA binding domain, a D-Cas9 or dead Cas9, if you will, that can be fused to various kinds of effector domains, such as transcriptional activators or repressors for transcriptional control, to DNA methyltransferases or histone-modifying enzymes for epigenetic modulation, to GFP or other fluorescent reporters to specifically label genomic loci. And finally, you can also even split Cas9 into its two constituent halves and fuse each half to chemically or optically inducible domains that dimerize upon blue light stimulation or chemical induction to regenerate the fully functional Cas9. This allows us to use CRISPR to actually achieve precise spatial temporal control to investigate dynamic biological processes. So there are so many things that we can do with CRISPR, really limited by your imagination and your ability to engineer these systems. But today I really wanted to highlight 4 different areas that people are really actively investigating. The first is the unbiased genome-wide profiling of Cas9 off-target mutagenesis. So the specificity of the CRISPR-Cas9 system is clearly important if you're interested in connecting particular causal genetic variants to their particular biological phenotypes. This, of course, also has tremendous implications for the potential translational applications of these programmable nucleases. Second, another great need in the field is the ability to efficiently but also precisely insert genes or modify the genome. As I alluded to before, although the homologous recombination allows us to do a lot of that, it's typically a much lower frequency pathway. So we'll discuss some efforts to improve this. Finally, I'll introduce the Cas9 knock-in mouse for in vivo disease modeling and discuss improvements in transcriptional activators for genome-scale transcriptional perturbation. There's a lot to go through, a short amount of time, but we'll try to get through it together. So, investigating Cas9 specificity. The field is kind of a black hole, as you can see here, right? Where CRISPR is presumably this nuclease that gets loaded onto the target locus of interest via sequence complementarity between the target DNA and the guide sequence of interest. So these are interactions that are presumably dictated by nucleic acid hybridization. So if you have this first order model of specificity, you might say, I should expect off-target sites throughout the genome to be sequences that have homology to my expected target site. So that might be 1, 2, 3, or more mismatches. So typically, Cas9 studies in the past, including my own, have computationally predicted these off-target sites and checked them in cells edited by Cas9 to see if you get off-target mutation. However, as Keith Zhang at Harvard likes to say, he likes to tell this story of this man, he's walking down the street, and he sees someone scrabbling on the ground under a lamppost looking for their keys. And so there's this man, he's a good Samaritan. He asks, why are you looking for your keys here? Did you drop them here? And they reply, no, I dropped them a ways back, but this is where the light is. So if we take this analogy and apply it towards trying to think about CRISPR specificity, how can we not look under these lampposts these computationally predicted off-target sites, but really try to illuminate more of the genome. And so if you back up and just ask, how do we figure out where these off-target sites are, and you lay out the genome into its constituent chromosomes, you might have a CRISPR-Cas9 complex that edits this particular site in green. And depending on various factors, that might be locus-specific, that might be governed by the chromatin state, that might be cell type or cell cycle specific, you'll get some number of off-target sites that are spread throughout the genome. Some of them might be predictable, but others may not. So how can we figure out what they are? So this study was done by Shengdar Cai, a postdoc in Keith Zhang's lab at Massachusetts General Hospital in Boston, took advantage of these biochemical methods to attempt to enrich for these off-target sites. And essentially, the assay is very simple and also elegant, where you take a double-stranded genomic DNA, well, the chromosome in your cells, and you introduce with Cas9 this double-strand break. However, the key is that you co-transfect in an oligo tag, which actually is exogenous sequence, so it's orthogonal to the genome, right? And this oligo tag will get introduced naturally by the cell at some unknown frequency into the break site. So if you then shear the genomic DNA, you'll get a mixture of sheared genomic DNA fragments, some that do not have and others that do have this oligo tag that you've introduced, right? And the idea is that any double-strand breaks that are introduced by the Cas9, whether on target or also off target, will get integrated. So then you can basically do next generation sequencing library preparation. So essentially you do end repair because you're not sure what kind of DNA ends you have after sonication. You do directional A-tailing followed by directional adapter ligation. And the idea here is you use these barcoded Illumina Y adapters that contain here a short barcode, a unique molecular index. And the idea here is when you have a random 8-mer, you can basically check before and after amplification so that if there are a lot of errors in your barcodes, you know that there's PCR bias. And you can essentially do error correction bioinformatically post-facto. But you have this pool of shared genomic DNA fragments with these Y adapters, and then you can essentially do specific PCR containing a priming site that is, primes on the oligo tag. Again, because the sequence is orthogonal, it should prime only specifically within the oligo tag that's in your library. If you do a second round PCR to add on the Illumina adapters, you end up with a final library containing the P5 and P7 Illumina adapters, the oligo tag that you inserted, and this sequence in black, the most important part of your library, where those off-target sites map to. So the slide header here is oligo tags specifically mapped to the Cas9-induced break sites. And what you see here is at 4 different sites in the VEGF-A and EMX1 loci, you can either 5' or 5 and 3' and protect these double-stranded oligos with phosphorothioate modifications. And the idea here is by adding in these base modifications, you can greatly introduce the half-life of a linear double-stranded DNA oligo tag inside the cell. Otherwise, it'll get quickly degraded by endogenous exonucleases and so forth. But what you find here is that if you end protect your DNA oligo tag, you get very specific and also high levels of integration. And this is simply a schematic showing that for your particular on-target double-strand break position, you can map these reads, these black sequences shown here, to that DSB of interest in both the positive and negative strand direction. And the idea then is if you take the first base of all of these reads and you map them to the, in this case, the on-target locus, you find that these first bases actually recapitulate very specifically the expected site of the double-strand break. That happens to be, for the S-paginase Cas9, 3 bases upstream of the PAM motif. And so where you expect the Cas9 to cut actually very nicely maps to where these oligo tags get integrated. So essentially this tells us we're getting very good and specific oligo capture at the on-target site, but what do the off-target sites look like? So for given target sites shown here at the top, you can basically enrich for a certain number of GUIDE-seq reads that represent presumably where those off-target sites lie. And so you can see some interesting features, for example, this non-canonical PAM, AAA. So previously in the literature, we know that Cas9 typically cleaves at NGG and NAA PAMs. However, there's also enrichment at these non-canonical PAMs that's something that we want to consider when we think about Cas9 specificity. The real take-home of this slide, though, is that not all off-target sites are equal, right? Some guides are more specific and more efficient than others. And for example, here, the HEC293 site 2, you see only 2 off-target sites, whereas this target here, the HEC293 site 4, has hundreds of off-target sites throughout the genome. And so this really tells us that if you have a particular guide RNA, you can do these kinds of experiments to try to screen them for specificity in your cell type of interest, but also that these methods of improving Cas9 specificity, such as double nicking, truncated guides, or dCas9 FOC1, and so forth, will also help you to get much more specific editing. And so then the next question is, does this work? Do these enriched GUIDE-seq reads actually recapitulate the level of off-target indels that we're getting at these predicted sites? And so what Shangar did was develop this creative AMP PCR, where essentially you do a single pooled reaction where introduced into the same PCR hundreds of gene-specific primers. And again, you have your library, you melt it, and then you introduce hundreds of gene-specific primers for the different genes that you want to check, essentially, right? Because you know where the off-target sites are, because you've mapped them here, you design a primer to each one, and then pool all these primers to your library to actually check if you actually got indels at those off-target sites. And essentially, you see here the cross-correlation for the VEGF-A site 1. It's actually pretty good. The R-squared is 0.81, whereas here, it's not quite so good. The VEGF-A site 3, the R-squared is only 0.5. So then the question here is, although here we are able to predict really well what the correlation between the GUIDE-seq read enrichment as well as the actual indels observed at those off-target sites, what is it that makes certain guides more, better predicted, if you will, than other sites? And that's something to do with the way the oligo tag gets integrated into the cell, right? Currently, the mechanism by which that integrates is kind of black box. Are there sequence bias? And is it biased by the chromatin state of that locus? These are absolutely efforts of further investigation. But the next thing that you can find that's pretty cool are actually detecting translocations. And so this is of utmost concern for people who want to make multiplex modifications with not just one, but what if you want to look at 2, 3, 4 or more guides? You might imagine if you make a cut here in the genome and another cut here, if you get a translocation event, you might really want to know and be able to detect what the frequency and the efficiency of those events. And so the key here, though, is because imagine that if you get the oligo tag ligated in such a way that to the left of this tag, you get a sequence from chromosome A, and to the right, you get a sequence from a completely different chromosome. You'll then be able to bioinformatically figure out that these two pieces are actually not in cis, but we actually find that they are. Therefore, this is a translocation event. And you can make these circus plots where for different guide RNAs, again, you can see that not all guides are equal. Some are very specific, like here for CF, whereas the VEGF-A site 2 actually has really large frequencies of translocation events. So how we can reduce or even increase the number of translocations, but also detect them, is something that will be required for the sort of further development of this technique. So that's GUIDE-seq, right? This is the unbiased genome-wide detection of off-target sites. What are the really next important questions? For me, absolutely, the first question is really, how sensitive is this method, right? Remember when I was saying that specificity of Cas9 is kind of a black hole, you kind of ask, well, where do I stop, right? Did I actually detect all the off-targets? How do I really know? And so experiments to really figure out how sensitive this technique is, how can we take into consideration these false negatives? So off-target sites that might be modified, but we didn't detect by this kind of biochemical enrichment. This is something we'll need to figure out. How scalable is this? Can we generate enough data points across different guides to try to figure out guide sequence intrinsic determinants of specificity? Are we able to pull out statistical patterns that are not cell type specific? Or even if we are, can we generate predictive algorithms so that you put in an exon of interest and then pull out all the guides and rank them by their off-target? That's something that I do in my tool, which you can find at CRISPR.mit.edu. This algorithm was trained on, I would say, sort of lower order models of specificity, whereas you might imagine experiments like GUIDE-seq would give you much bigger and richer data sets to train these kinds of algorithms. The real question is, how many data points can I get very easily? Finally, how does Cas9 specificity vary by the locus and the cell type? And again, the next question I want to ask is,
can I ask just a general question?
Yeah, of course.
We started that section off with the light, okay? And the question really was, I understand all the complexity, but from what your experience here is, how many off-target sites that are predictable? What's the percentage of those that actually get hit versus the ones you were looking for, which were out? What is that percentage?
Right. Well, so I guess if I'm understanding you correctly, if you look at
the beginning, the degree to which in the dark, i.e., things I would not have predicted would have been.
How well did that computational prediction work? Is that what you're asking?
How often do they occur in those sites versus your predicted off-site targets?
Yeah. The unsatisfying answer is that it completely varies. I would say that you do capture a subset of the computationally predict off-target sites. However, these experiments actually show you that there are a lot of off-target sites that you would never have predicted based on these lower order models of specificity. So basically what this really tells you is that these initial models actually didn't model CRISPR specificity very well. However, the other thing that I have to really emphasize is that there's a real disconnect between experiments that you might do in cancer cell lines versus modifications in somatic tissue in vivo, right? That we know the dosage of Cas9 and the length of the expression all really tunes its specificity. Here you're actually transfecting in massive amounts of these CRISPR reagents. If as you titrate this down, specificity of the system generally shoots up, and that's kind of intuitively obvious just by enzymatic concentration. But I suppose we just have to continue generating more of these data sets to really understand the patterns. Yeah. So this specificity, of course, is a really important, outstanding problem in the field. Another one is, how can we get efficient and precise gene editing, right? And so there's this sort of field of thought that there's a certain level of NHEJ and a certain level of HDR, and that we can somehow modify the relative levels of these two patterns, potentially to increase NHEJ so we can get higher levels of indels, or even to increase HR to get much higher levels of recombination for more precise types of mutations. So I basically have this slide here again to basically re-emphasize that non-homologous end joining is a high-frequency, error-prone repair pathway for the introduction of indels for gene loss of function, whereas homology-directed repair typically is the repair of broken genomic DNA with a sister chromatid. However, by introducing an exogenous repair template with homology arms that look like the sister chromatid, you can essentially hijack this pathway in the cell to insert your gene of interest, shown here in dark blue. But this is, of course, a very low frequency mutation. The basal rate is maybe 1 in every 10 to the 6 cells. With nucleus-stimulated HR, it can be as high as low percents or low fractions of percent depending on the cell type and the selection you use. But what if you want to do multi-site HR? What if I want to do in vivo editing? That kind of correction may not be quite sufficient. So these are experiments done by Sheng Ding at UCSF and Stanley Chi now at Stanford, and they performed a high-throughput chemical screen to try to look for small molecules that can improve homologous recombination efficiency. And so the very simple assay here is in mouse. They chose actually mouse ES cells because these are cell lines that typically have much higher basal rates of HR, and introduced into the nanog gene this homology donor containing in front of the stop codon, the last stop codon of nanog, P2A NLS SFGFP. It's just a very fluorescent, superfluorescent form of GFP. And if this integrates, you get this repair event where you, under the nanog promoter, you can drive the nanog gene, but also an NLS SFGFP. And so if you look at these box plots, you essentially have these controls where you get very low levels of background, but when you introduce the Cas9, your guide RNA targeting analog, and your donor template, you get roughly 17% homologous recombination. And then what they did was they developed a chemical screening platform where you took millions of ES cells, electroporated Cas9 guide and template, plated them out into 3 to 4 plates, plated on top a library of small molecules, and then used an automated imaging software to analyze GFP fluorescence in each individual well. And what they found was that out of 4,000 compounds that they screened at a basal rate of about 17 to 18% homologous recombination, which is read out by the proxy GFP positive nuclei, they found two small molecules, Burfeldin A and L7-55507. Burfeldin A is actually this inhibitor of intracellular protein transport between the Golgi and the ER, and L7 is this adrenergic receptor agonist. So kind of interesting known mechanisms, not really quite clear what their connection might be to homologous recombination. Nevertheless, they got a significant and reproducible improvement in the number of GFP nuclei. They also found these two thymidine analogs, interestingly, that actually also led to massive decrease in the rate of HR. And essentially here you have confirmatory FACS plots showing low background ES cells naked, the basal rate of HR, and improved HR about 2 to 3-fold in the L7 and Burfeldin A chemical compounds, and significant decrease with these AZT and TFT thymidine analogs. And so this works in mouse ES cells. They really wanted to ask how generalizable the system is across different human cell types. And so they actually designed into ACTA2 this smooth muscle actin gene, which is highly expressed in many cell types, and introduced in a homology donor with a very similar principle, just putting in T2A venus. And in HeLa cells and K562, so these are cancer cell lines, this is suspension cell line, and various kinds of primary cells, they found that they got a very consistent improvement in the homologous recombination rate across these different cell types, thus showing that this appears to really, these small molecules appear to act on a generalizable mechanism for improving homologous recombination. So this was all done with these more traditional double-stranded plasmid HR donors. And they wanted to see if using short single-stranded oligonucleotides, which are typically used to introduce point mutations, where they can also improve homologous recombination. And this is actually one of the most interesting points in the paper for me, where you see here the Indel allele frequency. They showed in the DMSL controls, but also treated with L7 molecule. They found pretty much the same levels of NHEJ. However, you got over an 8-fold improvement in the level of SSODN integration into the targeted site. So what this means for the interplay of NHEJ versus HR is kind of interesting, because what we typically find when we do these kinds of HR experiments is that your level of HR integration scales linear. Well, I'm not sure about linear, but it definitely is very positively correlated with the level of NHEJ that you also measure. But here they actually got really significant increase, but not in the Indel frequency. Then the final question they asked was trying to get at this interplay of NHEJ and HR, where if you look at these thymidine analogs that repress HDR, did they also enhance NHEJ? And so they took these ES cells that had a GFP mapped in and essentially used guide RNAs that targeted this GFP. And the idea is the efficient NHEJ will turn the GFP off. So they did FACS to look at the fraction of GFP negative cells. And what they found was that whereas the L7 small molecule typically led to a decrease in NHEJ, the thymidine analog had a very significant improvement in Indel frequency. So then, of course, the open question that wasn't really addressed in this report is, what is the mechanism by which these small molecules affect HDR and NHEJ? What are the longer-term consequences on genomic instability and chromosomal integrity? How do the cells choose whether to use NHEJ and HR? How redundant are they? What can we really do as cellular engineers to try to modify this, right? It's a difficult field because typically experiments done in cell lines are very useful, but in the DNA repair field, this is very difficult given their polyploid nature. But primary cells are difficult to handle, transfect, they senesce after a limited number of divisions. So this is really something of really growing interest in the field, absolutely. And finally, can we manipulate these pathways? Can we simply just modify the cells to actually get really efficient integration? Or is there actually a need for an orthogonal system, much like Cas9 for NHEJ? Do we need an orthogonal system such as a programmable recombinase, maybe a targeted transposase, for very efficient integration at your locus of interest? These are all things that I'm sure people are exploring. So I'll quickly introduce the Cas9 knock-in mouse for in vivo disease modeling. I'll introduce the neuroscience data in this paper, although there's actually a lot of interesting work done on modeling cancer mutations in the lungs of these mice as well that you can check out offline. So this work was done by grad student Randy Platt in Fung's lab at the Broad. And essentially what he did was to knock in Cas9 into the ROSA26 locus of these mice. And so you have this CAG promoter driving in a lock stop lock. So this is a triple polyase sequence flocks upstream of the Cas9 fused to P2A EGFP. And of course your classical positive, negative antibiotic selection markers. And so you knock this in and you essentially generate a line that's a Cre-dependent SP-Cas9 line. And then you can cross it with a beta-actin Cre driver line to get constitutively expressed Cas9 in these mice. And essentially, you see here these wild-type mice and the constitutively expressed Cas9, only the Cas9 expressing mouse has this EGFP expressed. And C and D are essentially panels that show neurons isolated from these mice constitutively expressing CRISPR typically have very normal electrophysiological signatures. Okay, and so you can also pair these, you can also breed these Cre-dependent lines with driver lines for tyrosine hydroxylase as well as palvalbumin. This is a subset of inhibitory interneurons. And the point here is that you get extremely good co-localization between the TH promoter as well as the Cas9. If you zoom in, you can see these particular individual cells that have really good co-localization of DAPI and nuclear marker, tyrosine hydroxylase and Cas9, whereas cells that have DAPI, no TH, also do not have Cas9. And you see the same pattern in these parvalbumin beta mice. Then if you do the in vivo editing, this proof of concept, you can package the guide RNAs. This is now significantly easier because you can get extremely effective and high titer virus just packaging a U6 guide RNA and a promoter driving your Cre recombinase. And you stereotactically inject this into the prefrontal cortex of the Cas9 mouse. And the gene that Randy targeted was highly expressed RNA splicing factor found specifically in neurons. It's called MUN or RBFOX3. And by deep sequencing of tissue punches from these edited Cas9 mice, you can see these very typical indels. And of course, by Western blot, when you add the guide RNA targeting UN, you get more or less complete loss of protein and very dramatic loss of MUN expression via immunostaining in these prefrontal cortex brain slices. So that's really the sort of largest scale physiology. What does it look like at the molecular level? You find here in the control mice, you have 100%, of course, MUN expression normalized, and about 80% knockdown when you inject the guide into a Cas9 mouse. You get over 80% indels in the MUN guides, but interestingly, if you look at the kinds of mutations found in these nuclei, you get almost, you actually get a vast majority of biallelic modifications that are actually both frameshift mutations. So this is actually very good, right, if you want to knock out a gene. Whether or not this really high level of frameshift mutation and also biallelic modification is, as neuronally specific or might be kind of a general mechanism in cells where you kind of edit to completion, if you will, well, it remains to be seen. So what's next? This is a mouse that's deposited at Jackson Labs, so you guys can get it. It facilitates editing in somatic tissue and primary cells. So one thing that they also demonstrated in the paper was transduction of primary immune cells derived from these mice with guide RNAs. So these are typically cell types that are extremely difficult to transfect and handle. And so by already having Cas9 expressed in these immune cells makes it much easier to do various kinds of downstream processing. You can also model multiple genes and mutations in biology and disease. This is particularly interesting for the cancer biologists out here who want to look at how driver mutations and tumor suppressors might interact and so forth. Finally, another interesting thing to look forward to over the next years are knock-in mice containing these Cas9 variants for transcriptional activators or epigenetic modifying Cas9 complexes to try to do in vivo studies of these epigenetic marks. And finally, one thing that could be tremendously exciting to do are in vivo high-throughput genetic screening as well. So the last piece I'll try to touch on briefly is the adaptation of Cas9 for high-throughput transcriptional perturbation. So in January of last year, Fung's group published a GeCKO paper using genome-scale gene knockout. But this paper, done by Silvana in Fung's group, engineered Cas9 to be a really robust transcriptional activator and also use this in the screen. And so if you think about what the challenges are in endogenous transcriptional control for modifying dynamic biological processes, you quickly find that at the single gene level, you require a really robust activator. So basically, you want to be able to target one guide RNA, so one Cas9 complex to this promoter of interest to turn it on. Similarly, at the network level, you want to do multiplex activation of genes or genome-wide screens. This really requires the single genes to not just robustly activate, but also be extremely reliable, because you may not be able to test all the individual guides for activity before you synthesize them in an oligo array pool. And so you require easy retargeting, robust activation, and activation reliability. So the previous generation of Cas9 activators, done by many groups, essentially was this very naive N or C terminal fusion of VP64, these transcriptional activator domains, to the Cas9 protein. And although this gave us really easy retargeting as opposed to previous generations of using finger or tail transcriptional activators, this actually still did not meet the requirements of robust activation reliability. Here's a typical gene trying to activate KLF4, and you find that you get very inconsistent activation across guides, but also not very high fold change. And so what we did was to introduce these MS2 aptamers onto the stem loops of the Cas9 guide RNA. And the key realization was that by taking the MS2 protein, which binds specifically to this aptamer, you can fuse on two more transcriptional activator domains from P65 and Heat-shock factor 1, and you could assemble this really the synergistically activated complex for very robust transcriptional control. And so you recruit not just the VP64, but multiple copies of the P65 and HSF1 domains at your locus of interest. And what you find here is that we get really robust activation. These are genes mined from the literature that had been previously shown to be very difficult to synthetically activate. And you can see here that the overall, you get over 100-fold greater activation than the previous generation, and that the activation was also extremely reliable. And so by tiling guides across different promoters, what you find is that in general, if your guide RNA is within the 200 bases immediately upstream of your transcriptional start site, you could find about 80% of your guides actually be functional. So that was a really important design principle for trying to do a screen. And finally, beyond single genes, you're able to activate individual genes by single guide RNA, but also simultaneously activate 10 genes by 10 guides, all transfected into the same cell. So here I want to introduce in a little more detail the concept of doing these genome-scale screens with Cas9. And the idea is you can synthesize a massive oligo array up to 100 or 200,000 different guides. And you can pool these guide RNAs into a lenti CRISPR plasmid and package it in pool into the lentivirus. And the idea here then is you do a low MOI transduction into your cell line of interest so that on average, one cell will have one infection event, and then you'll have multiple cells that actually didn't get any guide RNA at all. But you can select the antibiotic to enrich for the cells that have your guide, and then you perform your enrichment experiment, whether by drug or so forth, to enrich for your desired phenotype. And so the basic screen that we did was to take these A375 melanoma cell lines, which contain a particular BRAF mutation at the V600E mutation position. And the idea here is that there's an FDA-approved drug for late-stage melanoma called vemurafenib, or Plexicon PLX, that specifically targets these BRAF mutations. It's effective for over like 80% of melanomas. And however, these cancers, these melanoma cells quickly develop resistance, right? So we wanted to try to figure out what were the genes that might mediate resistance to the vemurafenib drug. And so what you can do is, looking at all the genes in the genome, take the guides, tile guides that are within the 200 bases upstream of the transcriptional start site, synthesize an oligo library, clone it into a lenti pool, and infect cells that were previously infected with the Cas9 P64 and the MS2 P65 HSF1. These are the components that we need to reconstitute the synergistic activation mediator complex in these cell lines. Then you have this library containing these guide RNAs, this real kaleidoscope of different genomes. Select with your vemurafenib drug, it's called PLX here, and then the cells that are resistant to the drug won't die, they'll actually be enriched. And then you can deep sequence these and look at the relative enrichment of the guide RNAs that you transduced to try to figure out which genes contributed to resistance to this drug. And what you find here is that you get very nicely consistent enrichment of your candidate hit guide RNAs. So for a particular gene, let's say EGFR, you see 1, 2, 3, multiple genes, multiple guides for the same gene of interest actually get enriched. So it's a very reproducible screen. And finally, if you look at the fraction of guides that are in the top 5% of enriched guides in your screen, about 80% of the guides get enriched, which is actually correlates very nicely with the finding that you can get 80% functional guides within the 200 bases upstream of the transcriptional start site. Finally, so we validated these hits, and essentially what you find is that the fold upregulation of the gene seems to correlate pretty nicely with the percent survival upon drug selection when you individually validate. And finally, that all these hits shown here clustered really well along known resistance pathways against vemurafenib, typically the RAS-MEK-ER pathway, the PI3K-AKT pathway, and the CMPK pathway. And so we found hits that were previously known based on cDNA ORF screens, that's shown here underlined in blue. But also we found these novel hits that might mediate novel mechanisms of resistance that weren't previously known. So with that, I'd like to really acknowledge my graduate postdoctoral advisor, Feng Zhang, the people whose work were highlighted in this talk, Randy Platt, Ophir Shlem and Stefan Konerman, Keith Zhang at Harvard, Sheng Ding and Stanley at UCF and Stanford, Editas Medicine, and all of you at MSU, especially Corey and Jamie for facilitating all this. Thanks very much. I'm happy to take questions.
Your activator, the previous section before this, you talked about how it was more effective when it was 200 base pairs upstream of the TSS. Mm-hmm. Was that contingent to the fusion protein itself? Do you think if you put on different activators, or
it's entirely possible? Yeah.
For this trans activator, for one promoter, how many guide RNA?
Only one. That was the whole point. So previously with these sort of V1.0 Cas9 activators, there were papers showing that if you actually tile a promoter of interest with multiple guides, you could get really synergistic activation, right? But the sort of the purpose of engineering this single complex was that only one guide would be necessary to activate your gene of interest.
I guess getting back to the off-target stuff, you had talked about how your algorithm versus other algorithms, but your programming itself. I was just curious, compared to Chop Chop from Harvard.
Sure.
They talk about how having an adjacent G to the PAM sequence would create a more specific target. Do you have anything to say on that?
Tesla, who did Chop Chop, is my friend. So I'll simply say that it was done in a very different context, and they're interested in zebrafish editing.
Yeah.
So I think it's still hard to say how generalizable these things are across different cell types and across different species. So the data I have was done in human cell lines, human cancer cell lines.
Yeah.
You'll probably get different results depending on what system you use. Cool, well, thanks very much. I'll turn it on to the next speaker.